Hi, my dear students. Myself Gurudev Mali, Assistant Professor, Department of Zoology, Government Science College, Chitradurga. Today we are studying BSc third semester Zoology syllabus on Karnataka LMS platform. My part is from BSc third semester Zoology, Physiology and Biochemistry paper. My part first unit. In that unit, it consists of 3 chapters. First chapter is Physiology of Digestion. In this chapter, we are going to discuss intracellular digestion, extracellular digestion, mechanical and chemical digestion of carbohydrates, proteins, and lipids. Also, the role of digestive enzymes, bile salts absorbed to area, and mechanism of absorption. What is meant by digestion here? Digestion is simply it is a physiological process in which large food molecules are broken down into smaller and absorbable forms. Here is, for example, we can take here is one example. This is the complex molecule now. This is polysaccharide, and then these polysaccharides are converted into monosaccharides, single units. This is called as catabolic process. Catabolism means what? Here is complex molecules are converted into simpler molecules. The same way here is digestion will take place. Here is complex food molecules are broken down into simple absorbable forms, that is called as digestion. Usually our food consists of protein, carbohydrates, lipids, nucleic acids, and vitamins also. These are essential for our body growth and development and also normal physiological functions. The different organisms have different form of digestive system based on their habitat and the level of organization. Already you know that the different levels of organization, cellular level of organization, tissue level of organization, organ level of organization, and organ system level of organization. According to their body organization, they have different structures in their digestive system which aid the digestion. The digestion may be occurs intracellular or extracellular. Intracellular digestion. Here digestion occurs within the cell, particularly in the vacuoles. For example, here is in case of amoeba. Here is intracellular digestion will be takes place when amoeba contact with the food material that extends their pseudopodia around the food and engulf the complete food material, and then resulting in the formation of structure called as phagosome, and all the digestive enzymes are released into this phagosome and digestion of food will be takes place inside this phagosome, and all the nutrients are absorbed in the cytosol and then undigested and waste materials which are released out by the exocytosis. In case of most of the protozoan sponges and some specialized cells in the multicellular organisms, we can see the intracellular. Next, that is extracellular digestion. Digestion occurs in the lumen of GI tract. In the sense, here is digestion takes place outside the cell. Outside the cell in the sense that is the lumen of stomach, lumen of intestine. See, here is this is the lumen of intestine. All the digestive enzymes are secreted into this lumen. By using those enzymes, here is the digestion of food molecule will be takes place. Hence, it is called as extracellular digestion. The human digestive system is the prime example for this type of digestion. It is digestive system of human. It consists of mainly two main structures. One is alimentary canal and second one is associated glands. Associated glands are also known as accessory glands. Alimentary canal, that is start from mouth and extend up to the anus here. Accessory glands or associated glands include salivary glands, liver, and pancreas, which supports the digestion of food. In this session, we are going to study the structures of alimentary canal. Here, first one, buccal cavity. The buccal cavity contains the tongue and teeth. These are very important structures for grinding and mixing of food. Teeth are responsible for chewing of food called mastication, and tongue is responsible for mixing of food with the various secretions of salivary glands. And after here is grinding and mixing of food, this food is converted into soft ball-like structure called bolus, which is enter into next part of alimentary canal called pharynx. The pharynx is common passage for both air as well as food here. Let us see. Here is another structure that prevent the entry of food into the trachea. That structure is called as epiglottis. Here is how the movement of food from pharynx to esophagus we can see by this animation here. How the epiglottis work here. You can see this one. Once again we can see here epiglottis closes the structure known as that is wind 5 here, and that prevent the entry of food into the wind 5 here. Next one, the next structure. After air is put passing through the pharynx into the here is long tube called as esophagus. It is about 40 centimeter long tube. It contains two sphincters. One is the upper esophageal sphincter and another one is the gastroesophageal sphincter, or we can say cardiac sphincter, and that controls the entry of food into stomach here. Here is movement of food into the in the esophagus is by the peristalsis process. Here we can see how the food movement takes place in the esophagus. Here we can see in the animation. Here is wave-like movement takes place. Here is contraction of longitudinal muscle and circular muscle antagonistically. Antagonistically in the sense when the contraction of circular muscle takes place, here is relaxation of longitudinal muscle will be takes place. This leads to the wave-like movement of food in the esophagus, and this food reaches to next structure of alimentary canal, stomach here. This stomach consists of fundus, body. This green region is called as cardiac region, and then this is called as pyloric region. Pyloric region consists of another two areas. One is pyloric antrum and another one is the pyloric canal, and totally this is called as pylorus, and it also contains two sphincters. One is upper, that is the gastroesophageal sphincter between the esophagus and cardiac part of the stomach, hence it is also known as cardiac sphincter, and the lower one that is called as pyloric sphincter, or we can say gastrointestinal sphincter, which is present between the stomach and duodenum of small intestine. Let us see the structure of stomach. Stomach consists of four layers here. Outermost layer is serosa, and the next layer is muscularis externa. It consists of three layers: the outer longitudinal muscle layer, circular muscle layer, and oblique muscle layer. Oblique muscle layer is unique to stomach. It is not present in the intestinal region. It is only present in the stomach region. And next layer is the submucosa. It contains various blood vessels and nerve plexus, and innermost layer is called as mucosa, and mucosa has various irregular folds, and this is called as gastric rugae. And then in that gastric rugae we can see, this is what we can see, gastric fit, and then at the depth of the gastric fit we can see the most of the gastric glands which secrete various enzymes and other factors which are responsible for digestion and also protection of the stomach layer. Here we can see the various gastric glands here. This is the cell called as parietal cell, or we can say oxyntic cell, that produce the gastric acid, that is HCl in the stomach. Their pH about here is 1.5 to 2 in the stomach here. Hence, there is a chance of damage of inner wall of stomach maybe takes place due to this harsh acidic nature. Hence, the another cells of this stomach called mucous neck cell that produce the mucus as well as bicarbonate ions which protect from this acidic nature. And next type of cells are called as enterochromatin like cells that produce the histamine. And then another type of cells are called as cheap cells, or we can say peptic cells, or we can say zymogen cells, which secrete a inactive enzyme that is called as pepsinogen, and another enzyme called as gastric lipase, which are helpful for the digestion of protein and lipids respectively. The next type of cells are called as T cells that produce a hormone called as somatostatin hormone, which inhibit the secretion of gastric acid. And next very important type of cells are known as gastrin cells, or we can say G cells, which produce the gastrin hormone which stimulates the secretion of gastric acid. Next part of alimentary canal is intestine. Here is this one is the small intestine and large intestine. First we are going to study here small intestine. The small intestine consists of three parts here. One is this is the C-shaped, or we can say U-shaped duodenum portion, and then this is the jejunum portion, and last, that is highly coiled portion, is ileum portion. That ileum is opens into the large intestine by ileocecal valve. Small intestine, it consists of four layers. The outer is the serosa layer. This is the serosa, and then next layer is muscularis externa. It consists of two layers: outer longitudinal muscle layer, inner circular muscle layer, and next layer is submucosa. It contains various glands and blood vessels also, and then the innermost layer is mucosa. This mucosa contains lots of circular folds, and these are called as plicae circulares, or we can say walls of Kirkring. If we zoom out this portion, we can see various finger-like projections. These are called as villi. Main significance of villi is to increase the surface area for absorption. At the top of these cells are called as enterocytes, and they have microvilli, and that is look like a brush border, hence it is called as brush border area, or we can say these cells are known as brush border cells, which secrete various enzymes for the digestion. And also it contain another type of cells called goblet cells. These goblet cells are responsible for secretion of mucus. And then here is at base of this villi, we can see here crypt of liver cone. This structure is called as crypt of liver cone, and that contain various liver cone glands, and their secretion helpful for the digestion as well as maintenance of inner wall of this intestine. The cells of Paneth, these cells are very important because of which secrete antimicrobial agent which kills the microbes which are enter into our body. And how can we differentiate the different part of small intestine? If you see the structure of duodenum, the duodenum has, here is a Brunner's glands at the submucosal layer, and hence it is called also known as submucosal glands or duodenal glands. And also crypt of Libercun is very simple in the duodenum region of small intestine. If you take a jejunum and ileum part, we can see crypt of Libercun. They are well developed with the Libercun glands like a Vanich cells. And then next one, ileum part. Apart from it having crypts of Libercun, it has a structure known as Peyer's patches. It is the lymphoid tissue present only in the ileum portion. Hence, we can differentiate all the structures from ileum by the presence of here is Peyer's patch. Next one, large intestine. Here is small intestine, that is the ileum portion which opens into the cecum part of large intestine through ileocecal valve. The large intestine consists of, this is the cecum region and then colon region, and here is a rectum region. Here is first region, this is called cecum, and then cecum leads to the colon region. The colon can be divided into ascending colon, transverse colon, descending colon and sigmoid colon. And then these are the compartment formed by the muscles, that is called as a haustra, and then which is helpful for the bowel movement of food inside the large intestine. And also this is the rectum part which stores the undigested food material, and then later it is agated by us. This is the anus part which is helpful for the agation of undigested or waste food materials. It contains two sphincters. One is internal anal sphincter and external anal sphincter, which controls the agation of undigested food molecule. This is regarding the alimentary canal of human digestive system. In our next session, we will going to discuss about accessory glands of digestive system, or we can say associated glands of digestive system. Thank you one and all.